All right. Let's aim to get started at 10:03. And then on the screen, you'll see a QR code for our event feedback form. So if you want to queue that up so you can give us a, fill out our form for us, let us know how we did, that would be great. Okay, well, it's 10:03. Welcome everyone to the Black and Comp Bio seminar series. Today we'll be joined by Dr. Tigist Tamir, and she's going to be giving us a presentation on her research. But first I'll introduce myself. I'm Melissa Minto. I'm the programming coordinator for BWCB, and then we also have Janae here.
Hi everyone, I'm Janae and I'm the director of BWCB.
Yes, okay, so we'll be your moderators today. If you have any questions, yeah, go ahead and put in the chat where you're coming from. Hello from University of Florida. I'm from North Carolina. I'm in North Carolina now. So I'm gonna go ahead and get started and introduce our speaker, and then gonna hand it over. Okay, so Dr. Tigist Tamir is originally from Ethiopia, and she's developed a passion for science during her teenage years. She pursued a STEM journey through undergraduate research at the College of William and Mary, and through programs through HHMI EXROP and NIH Prep, where she explored areas such as developmental biology, cell signaling, and cancer biology. Dr. Tamir earned her PhD in pharmacology at the University of North Carolina, Chapel Hill, focusing on kinase-mediated regulation of oxidative stress response. In her current work, she uses multidisciplinary approaches involving biochemistry, multi-omics, and computational models to investigate how cells regulate oxidative stress within complex signaling and metabolic networks. Committed to promoting diversity in academia and research, she mentors students through various programs and fosters inclusive peer communities while sharing a diverse scientific stories through her video miniseries, Edges and Notes, to inspire and build an inclusive scientific community. So join me in welcoming Dr. Tigist Tamir as she gives us a little peek into her research.
Thank you so much for the lovely introduction. Let me quickly share my screen. And hopefully everybody can see. All right.
That's good.
Yep, it looks great.
Great. Thank you everyone for joining us on this early Monday morning. I appreciate you taking the time to be here and listen to some of my research progress. Today I'm going to be talking about triple negative breast cancer, specifically our efforts towards finding new targetable molecules and also defining new signatures that potentially can yield good therapeutic outcomes. So a little bit introduction into triple negative breast cancer. It is characterized by the loss of 3 different receptors, the ER, PR, and HER2 receptors. And loss of these receptors leads to increased proliferation and oncogenic potential, and TNBC accounts for approximately 15 to 20% of all reported breast cancer cases. And one of the actually motivating factors, there is my study, TNBC is actually, it's more prevalent in Black and Hispanic women, especially under the age of 40, which limits lifespan and affects kind of presents a health disparity risk for Black and Hispanic women. And one of the challenges with TNBC has been that it's quite a heterogeneous disease, meaning that the treatment options are limited, but also that most patients, as they come into the clinic, they are given chemotherapy as a first line of therapeutic intervention, and that usually leads to resistance that cannot be overcome after the fact. So it's our goal that we define new targets and potentially new therapeutic approaches that could yield better patient outcome. And one of the ways we do this is by studying signaling pathways. And a quick introduction for those who are not familiar with cell signaling, it's a process of pretty much relaying information from external and internal cues where, let me make sure that I have the pointer here. So you can see the pointer, great. And the way signaling pathways work is, there we go. Okay, cool. The way signaling pathway works is that there is, for example, an external cue, such as a growth factor that binds to some sort of receptor, or there's an internal cue where there's turnover of signal that leads to response. And in the case of cancer cells, usually these signals promote proliferation, metastases, things that make the cell quintessentially like dividing uncontrollably, as well as metastasizing to new locations. And if like you want an analogy, one way to think about this is, if you've seen the movies Mulan, signaling pathways are where basically on the wall of China, on the Great Wall of China, there is a fire signal that goes down the wall and alerts the country there's an incoming invasion. So that's how signaling pathways usually work, where there is signal being transmitted, leading to more proliferative capacity for cancer cells. And one of these signaling pathways modalities we study is phosphorylation of proteins. This is a post-translational modification of proteins, and it's quite reversible and dynamic, where phosphorylation moieties are added by kinases onto proteins, and they can be reversed by phosphatases. And these enzymes pretty much regulate the expression, localization, and function of these particular of proteins in the cell. And most phosphorylation events take place on serine, threonine, or tyrosine residues. These are amino acids that are most commonly phosphorylated. And this, sorry, I have to like keep moving the attendant bar. Anyways, this phosphorylation events leads to signal transduction downstream of the kinases or phosphatases to yield the phenotypic outcomes we usually see with cells. And we particularly study tyrosine phosphorylation, which is most well known for being downstream of growth factors that are known promoters of oncogenesis. And in previous studies from our lab, we've shown that we can actually take patient samples from histology banks that are usually taken like when there's routine biopsy of suspicious lesions. And these these tissue banks allow us to profile the type of signaling events that are happening in patients. So we've developed a quantitative way to measure what kind of signaling pathways are activated in patient samples. And then we went ahead and actually have evaluated which targetable nodes we should focus on, especially for triple-negative breast cancer, where we've shown that in xenograft models, which means that these are tissue that gets transplanted into mice. And in small-scale studies, we've shown that targeting a specific family of kinases, known as Sark family of kinases, actually yields better outcome or tumor reduction in mouse models. So one of the things we asked was, well, now that we've developed this technology, can we evaluate tissue samples from patients as they come into the clinic and determine if we can suggest any new therapeutic avenues? So can we optimize TNBC treatment options and define patient-specific molecular signatures so that as patients come in, they don't all go directly onto chemotherapy? If there are other options that they can consider, that physicians can consider, we should give them the option to kind of save lives. So the way we did this is in collaboration with the Mayo Clinic Breast Cancer Spore, physicians at Mayo Clinic sent us fixed patient tissues from 150 patients. And we went ahead and extracted protein from that. And what we do is we take these extracted protein, we chop it up into smaller bits, and then we label it with different isotopes so that we can do a multiplexed pull down of these phosphorylated proteins, and then use mass spectrometry-based proteomics to identify how much of that particular phosphoform of protein is present in each sample. And we do this in a kind of scaled version where we can multiplex quite a bit of sample at the same time, so we can look at the different amounts of these phosphoproteins across different patients. And what that allows us to do is we can now define which signaling pathways are altered in which group of proteins. And then we can take that information and then divide patients into groups where we can suggest perhaps, the response for this group is going to be perhaps suggesting like immunotherapy instead of chemotherapy, or other neoadjuvant treatment options. So by doing this type of sample processing and mass spec-based phosphoproteomics, we profiled 150 patient samples, and I'm showing you here a heat map of Spearman correlation analysis, where phosphorylation sites are co-correlated and showing different clusters of phosphorylation sites on proteins that we identified in these 150 patients. And we can actually do a similar type of co-correlation analysis for the samples where we can cluster patients based on these phosphorylation signals and suggest which ones actually have a differential expression of these phosphoproteins.
Morning in progress.
Oh, sorry, was that a question? Okay, cool. And if we look at the hierarchical clustering analysis of these phosphoproteins shown here on the vertical column with patients going horizontally, what we start seeing is actually different clusters of phosphoproteins. One group is involved in RNA processing, so proteins that are known to regulate RNA splicing or processing, or protein translation machinery, were differentially phosphorylated in some groups of patients, as you can see here, but more than others. And then another group was we saw innate and adaptive immune response in some patients that is highly upregulated versus in other patients where it's quite down. And then as we have previously identified, we show that there are a specific cohort of patients that have elevated Src family of kinase signaling or receptor tyrosine kinase signaling, which are all known to promote oncogenesis, or are drivers of proliferation. Just to corroborate our previous results that have been done on just mouse models, we show that the Src family kinase signaling we observed, these substrates actually drive cell-cell communication, specifically those associated with adhesion and migration. And this actually becomes essential for triple negative breast cancer because most of the time, these signals drive the metastasis observed in patients. So our initial study that suggested that perhaps this is a targetable pathway for patients that do not respond to chemotherapy or have metastasized cancer. It actually, we can get that kind of information from pre-treatment samples, which was kind of like ensuring the applicability of this technology. And then the next cohort of patients were those that had elevated or downregulated innate and adaptive immune signaling. And one thing to note is especially recently with the excitement over giving patients immunotherapy, we have been able to define patients as having a cold tumor, an immune cold tumor, or an immune hot tumor, meaning that in the immune cold tumor, there is little to no immune infiltration. So T cells that usually kill cancer cells cannot go into the tumor core and start dismantling the tumor growth by killing cells. Whereas in an immune hot tumor, T cells are able to infiltrate. There is less immunosuppressive signals in this tumor, and by being able to infiltrate, the T cells can recognize cancer cells and attack that cancer cell to lead to cancer cell death. And by introducing immunotherapy that blocks immunosuppressive signals, in multiple cancers, studies have shown that there's efficacy, and this is has not been explored sufficiently in triple negative breast cancer. So we were very excited to kind of see this differential regulation of immune signaling in some cohorts of patients. So what did we see in our samples? So we can definitely see that there is immune hot or immune cold tumors. So these this is a heat map showing different phosphorylation sites on a T cell receptor, which is the receptor that recognizes cancer cells and directs for either immune-mediated apoptosis or other ways of killing cancer cells. So we can see that these CD247, CD3D, and CD3E are proteins that make up the T cell receptor. And you can see that there's differential phosphorylation where these group of patients have lower levels of phosphorylation for T cell receptor, while this group of patients have high phosphorylation. And if we actually contextualize this and look at where these phosphorylation events are happening, and this is a diagram of the T cell receptor where there is CD3 flanking the T cell receptor. And in the intercellular domain of the T cell receptor are these phosphorylation sites that are known to be phosphorylated. And when they're phosphorylated, they actually lead to downstream T cell receptor signaling activation and intercellular localization. And that way they can they can recognize cancer cells and induce an apoptotic phenotype in cancer cells. And what we're actually seeing is that there's patients that have high receptor signaling where perhaps an immunotherapy treatment would be more efficacious compared to just chemotherapy, whereas some patients might not be responding to immunotherapy. And if we look at enrichment, the enrichment score, or like the overrepresentation of this pathway, we can see that for the majority of patients, we're seeing this downregulation of T cell receptor signaling. So if we were, and these samples actually came to us after the study was done, but if we were in the clinic at the time when these samples were biopsied from patients as they were coming in, we would suggest the group of patients that would most likely respond to immunotherapy based on this information. And if we actually look at individual patients, so the goal here is not just to find the signature of or the group of patients that will respond, but also give the patients an option for some precision medicine and personalized medicine, where if we look at each individual patient, we want to be able to say, okay, so this patient has downregulation of this adaptive immune response, but there are other patients here represented as squares that have high adaptive immune response. And one of the things we've been doing is group patients into high responders and low responders for specific signaling pathways. That way we can target the specific pathways where the patients actually would be the most responsive. And then another kind of surprising thing we found was phosphorylation on these protein translation and RNA processing regulation machineries, particularly these proteins called SRSFs, which is kind of a new subtype. And what SRSFs do is they, when phosphorylated, actually lead to downstream regulation of RNA splicing, where the phosphorylated form of SRSF proteins leads to differential mRNA forms that could be actually essential for T-cell receptor signaling. One of the links we made is that if we are getting this phosphorylation event where this alternative splicing of mRNA is highly upregulated, and we can identify the patients that have this elevated mRNA processing signaling signature, we can actually overlay the signals we observed for the T-cell receptor signaling versus this RNA splicing machinery proteins. And what we see is that an opposite phenotype in some patients where we see in some patients where there is elevated phosphorylation of T-cell receptor signaling, shown here in green, while in yellow, while the signals in green for the RNA splicing machinery are elevated. So they're showing an opposite phenotype where there's alternative splicing of mRNA with a downregulated T-cell receptor signaling. And in various groups of patients, we're seeing that there's an opposite phenotype for most of these signals. And what this brings into question is, well, alternatively spliced proteins can actually be presented as antigens or the small peptides that are recognized by T-cell receptors. So perhaps this alternative splicing cohort of patients that show this upregulated signal can be the potential recipients of immunotherapy, because they might respond better because they have more antigens present on the cancer cell surface. So as we go through each patient's molecular signature, what we were able to identify is that, okay, this patient has low immune suppressive signal. So these are a group of signaling pathways that are markers of immunosuppressive, meaning that there is less immune response in the tumor core. And we see that these patients have high RNA splicing features or regulation of RNA splicing, but they're not immunosuppressive. So if we treated like this patient, for example, with some sort of immunotherapy, we can actually get better results than with just chemotherapy. Oops. Oh. Great. So what I've, oh no. It keeps going back. Okay, what I've shown you so far is that triple-negative breast cancer patients can be stratified by their phosphotyrosine signaling signatures. And this can help us potentially optimize the therapeutic avenues that clinicians take as patients come into the clinic for biopsy to confirm whether they have cancer or not. And one of the things we've previously identified is Stark family kinase signaling activation in some tumors, but we also now have another signaling pathway, which is the immune cold or immune hot tumors that's not me, that also show regulation of RNA splicing, that we can potentially find new therapeutic intervention for. So these cohort of patients that have this upregulated or downregulated RNA processing machinery or protein translation machinery can be recipients of new types of targeted therapy. So the goal after this is to kind of generate an integrated model to predict better therapeutic avenues. So what we'd like to do is, by getting these biopsies, compare the information. Usually there is genomics, transcriptomics, epigenomics analysis that is done on these patient samples as they come in. And we want to get that information, look at the different stage of the phenotype. Is it like a more severe disease versus is in the early stages? And then identify what type of treatment, first line of treatment these patients were given, and then be able to predict survival or disease progression based on the signaling pathways, and by identifying which patients would benefit from better treatment. And another thing we are considering is that as patients progress through their different therapy cycles. We want to be able to predict response and also adaptive resistance to the current line of therapy. The one challenging aspect of cancer treatment is that regardless of what kind of new therapy we come up with, there is always a potential for adaptive resistance. And what we want to be able to do is that as we're giving them whatever the first line of therapy is, we want to be kind of thinking about what things can arise from that drug induced adaptive resistance, and then be able to knock that next pathway down so that patients can remain disease-free for a longer time into remission, and also prevent relapse. And as we move forward, we want to be able to differentiate between which types of treatment options are available for patients, and be a little bit more precise. So this is the precision medicine aspect of using omics technologies, but be a little bit more precise as to what kind of therapy patients get so that we save lives in the long run. And this could also help us identify which patients don't need to be on therapy for an extended period of time, where we can intervene with surgery or radiotherapy after targeted therapy, so that we shrink the tumor first, and then we remove as much of it as possible, so that the patients enter disease-free as opposed to remission. And with that, I would like to thank everybody from the White Lab. Forrest has been a great mentor and has done a fantastic job collaborating with this amazing physicians at the Mayo Clinic and the Breast Cancer Spore group, where they have been kind enough to share all of this patient tissue samples, as well as a lot of the data they've collected on the genomics side for us to be able to do this type of work and identify new ways we can treat triple-negative breast cancer, and also my funding sources. And with that, I'll take any questions.
Thanks. That was great. Yeah, whoever's clapping in the background, that was so great. If anyone has any questions, feel free to unmute and ask them, or put them into the chat and I can ask them on behalf of you.
Hi, that was an absolutely wonderful presentation. Triple-negative breast cancer is a topic that is near and dear to my heart. That's what I did my dissertation on. So, and right now I switched to doing work in computational modeling. So I was completely all in for your presentation. So yeah, it's awesome. So my question is, and I may have missed this and you may have explained it, but I may need a little bit more explanation. One of the things I'm interested in is, as I'm learning this field, new ways to use computational models. So can you explain a little bit more about how the computational model fit into this particular research and how you built it? Was it an ODE model? Was it Boolean, et cetera?
Yeah, so that's an excellent question. So what we are currently doing is actually not an ODE model. We're like doing a bit of shallow machine learning, so doing unsupervised learning and supervised learning based on labeled patient samples. So basically, especially like if we get like histology information, and one of the things we're currently working on is trying to look at the immune infiltration and trying to understand if the signaling pathways we see predict the immune infiltration level. So one of the ways we're doing is like doing a lot of unsupervised learning of PLSR, PLSDA type of models that can do prediction. And in terms of like a more dynamic form of prediction, we can't do that with like patient samples, unfortunately, because you can't do a lot of perturbations, and that requires doing like cell culture based models that you can specifically alter different parameters. So we haven't gotten there, but what we're doing is more of the machine learning approaches.
Got you. That answered my question perfectly. Thank you.
Thank you for the question.
I had a question about, I do some work in triple negative, and I'm really interested in the disparities that we see in survival as it relates to triple negative. Even when matched by stage, grade Black women for some reason have lower overall survival, recurrence-free survival, and progression-free survival. What do you think could be contributing to some of those differences? And have you thought about, or have you found if there's a particular type of triple negative, or if there's a particular signature that are found in women of African descent that may be driving those differences?
Oops, sorry. Oh. Okay. Thank you for the question. We haven't found, most of these patient samples we get are de-identified, so we haven't found something that, like at least from our data set, that indicates a particular difference between African American patients versus others. One of the things we're currently actually working in collaboration with other clinicians from Colorado and UCLA is actually looking at postmenopausal breast cancer and postpartum breast cancer. And in those cohorts, we've been able to identify specific signatures of activated pathways, particularly the Src family kinase pathways. So we were not sure if that is driven by mutation or by the type of tumor microenvironment present in those cancer types. So that is still an ongoing thing we're exploring. But so far, we haven't been able to pin down specific molecular signatures that would be associated with the higher risk TNBCs in Black women.
And you mentioned the microenvironment, and given how obesity is now increasing, or is now being a risk factor for so many diseases.
Mm-hmm.
I wonder, how obesity is, and diabetes and cardiovascular disease and these inflammatory disorders could be potentially driving those differences in outcomes. So I have a lot of interest in that, and we just started doing some spatial genomics, which is really cool. I can't wait to see what the results of that research yield.
Yeah, no, that's actually an excellent point. There's another side of my work, which is focused on obesity and its contributions to cancer. And one of the things we've worked with other collaborators is if we have actually an obese extracellular environment, an obesity particular extracellular environment, whereas a lot of nutrients coming from fatty acids, and saturated fatty acids in particular, we actually see a differential response in metastasis, specifically in triple negative breast cancer cell lines. This is in cell line based models. We see a differential response depending on whether it's an obese microenvironment versus a lean microenvironment. So that's something we're actually exploring. And I guess I'm not an epidemiologist, but one of the things to consider is the high correlation between metastasis and obesity, specifically for these types of cancers. So it's super exciting. I think there's so much potential in exploring further in this field. We're just scratching the surface.
Good morning. That was an amazing presentation. Thank you. A clarification question first. So the cluster with the hot tumor patients is the same with the one with the elevated mRNA splicing events? Is there association between hot tumor with the, so it's the same cluster, or is
it's the reverse?
Okay, it's a
negative relationship where the hot tumors have low mRNA splicing and the cold tumors have high mRNA splicing, which kind of.
But you mentioned that the elevated mRNA splicing events might indicate a good outcome after immune therapy, right?
Mm-hmm.
But this is weird, isn't it? If the tumor is a hot tumor, it means that you have immune cell infiltration, so you would expect to have a better outcome with immunotherapy.
Yes, but that is assuming that there is no immunosuppressive signals as well.
Uh-huh.
So one of
Okay.
the challenges of immunotherapy is that like macrophages could be polarized differentially. They could be immune suppressive or immune activating.
Mhm.
So I think one of the challenges so far has been trying to make sure that there is no immunosuppressive signals in the hot tumors.
Mhm.
Yeah.
Okay, that's very interesting. And one last question. So do you think that it's the different inflammation or response events that lead to the differences in the mRNA splicing events, or it's vice versa, the mRNA splicing events that lead to differential tumor response and different inflammation, inflammatory environment?
That's actually an excellent question. Thank you so much for that. I think it's like a chicken or the egg question.
Yes.
Because if initially the cells are, in this hypoxic environments, growing as a tumor nodule, you would think that there will be like more inflammatory signals that should promote, immune infiltration.
Mhm.
But one of the things, people have shown in the tumor microenvironment type of studies is that that also has to support T cell, for the T cells not to die, because that environment is very noxious. Because the more hypoxic it is, the less T cells are going to be able to infiltrate and survive. So it's a chicken or the egg question. Do you inhibit inflammatory response in order to promote to immune infiltration? But then that has other consequences as well. And I think there have been some clinical trials now that have shown that if you do if you give patients immune therapy, they actually have off-target inflammatory responses that actually are detrimental for the patient. So I don't think we've actually reached a point where there's like a perfect balance of the two. I think that's something that we have yet to figure out.
Okay, thank you.
Thanks.
Hi, Jesus. That was also a very, very nice presentation. I also have a question, and I may have missed one or two slides, so it's possible you mentioned it. So whenever we see this kind of work, we always want to put it in context of what we already knew. So I'm curious if you had gene expression, mRNA expression data on the same samples and looked at the coincidence of your protein-based sort of subtypes with the known TNBC subtypes.
Yes, so we did have mRNA transcriptomics data from these patients. One of the things we noticed is that our the clustering based on our data actually did not align with the clustering analysis that was done on the transcriptomics data. So we think that in terms of the subtyping, I don't think our data fits into the different subtypes of TNBC. That usually happens with either histology or with transcriptomics data. We're seeing patient clustering by purely the signaling modules that are altered across patients. So and the other thing to consider, and this is quite controversial, is that we do think that the type of signal that's there is not necessarily entirely dependent on protein expression. Obviously, proteins have to be expressed to be phosphorylated and to have differential functions, but we don't think that it's entirely present, it's entirely dependent on the expression levels. If you have a certain pool of unchanging of a certain pool of the proteins, the specific protein being phosphorylated, and that doesn't change regardless of the expression of the protein. So that's something we're working on. It's obviously a controversial point because the central dogma suggests that there's more expression and then that leads to more function, but some proteins have to be regulated post-translationally. So that's something we're working on.
Okay, thank you.
Thanks.
But yeah, if you wrote this up, you would definitely get that question from a reviewer.
Yes. Yeah, we're thinking more and more about that. We've actually processed the global just protein expression of these samples as well to make sure that we address that kind of points. So thank you.
I have a question about the samples. So, because you saw these subtypes that it made me wonder, were these biopsies collected at like similar stages of cancer? And if so, or if not, like how do you control for that within the analysis?
Yeah, so these samples were not collected at the same stage because you can imagine as patients come in, they could be at different disease stages. What we're actually taking that into account in some of our downstream analysis is if like the different stage of the tumor can be associated with the type of signaling it's undergoing. So far, we haven't been able to differentiate because also this is like the first biopsy that patients like receive, like the first biopsy as they come in. So it's not even the biopsy of the tumor after treatment or like throughout treatment or post-treatment. So what would be like really exciting is to try and track patients and get samples from the tumor at different stages as they go through the treatment phase.
Thanks.
I have a quick question about the splicing activity. I know you mentioned the hot and cold tumors have different profiles, and you mentioned the SRSF phosphorylation being associated with upregulated splicing. Do you have an idea of what types of splicing events those are, and maybe the distribution of that activity?
Yeah, so the quick answer is no.
Okay.
The long answer is we have it, at least from the transcriptomics data, we don't have a deep enough read for us to differentiate if there are differentially spliced forms of any protein, honestly. But one of the things that SRSF mediated splicing has been associated is like differential splicing of oncogenes like MYC, which might yield differential response to chemotherapy. So the way we're thinking about it is maybe with the global proteomics side of the analysis, we might be able to identify the differential splice forms of the proteins, which is going to be tough. But the other thing is, if we get deeper reads from these samples, perhaps we would be able to tell what things are differentially spliced. So yeah.
So by how not deep are we talking? Like
the generic transcriptomics analysis is what has been. I think you'd have to be very particular and selective if we want to try and figure out what things are differentially spliced.
I see. So yeah, I guess that's connected to my other question. Other types of RNA processing events like NMD or things like that, is that within your awareness or maybe not the focus of the...
Yeah, so not from this data set, but we have like from the same samples, we've done other like more global phosphoproteomics analysis, not just tyrosine phosphoproteomics analysis. And those we actually do see the other forms of splicing machinery being phosphorylated. And we're trying to do correlation analysis between that and their expression, trying to figure out if that is differentially regulated across patients. And if it is, does that align with SRSF phosphorylation that we already saw?
I see. Cool, thank you.
I have a question about once we have the ideas about which particular pharmaceuticals will work best for these particular patients, will there be more work in determining, okay, what dose should this be given at, more of the pharmacological aspect of it?
Yeah, so I think for the clinicians at Mayo, usually when they have like the cases that are particularly difficult to treat, they just go down the line of potential treatment options. So the goal now is to, as patients come in, do this type of analysis, identify potential targets, and use already approved drugs in order to kind of circumvent having to like figure out what dose, what type of pharmacological agent needs to be used for treatment. So using already approved, like the goal of some of this work is trying to repurpose drugs pretty much that have already been approved for other treatment options, which is why I think the immune therapy angle is a little exciting to us, because we already know what's been approved out there, what is being used for other types of cancers, and perhaps that's something we can repurpose for this type, because I think approval for most drugs takes like a very long time to make it through preclinical, clinical, different phases of clinical trials. So we want to be able to like give these patients an option that is immediately applicable.
It makes perfect sense. Some of the work that I do is working on computational pharmacological models to even further personalize the drugs that have already been approved to a particular patient. As we know, one of the problems with the approval of the drugs is that most of the people that have been in the clinical trials don't look like us. That they're not the same gender, they tend not to be the same race, if that's a word that you want to use, don't have the same ethnic background. So often, some of the pharmaceuticals may react differently, right? So I'd be curious to see if that would be something that might be useful to do, to have some kind of model that can say, okay, based upon what we've seen from reactions to this particular drug, we see that people with this particular profile, metabolic profile, for example, and like like it was previously talked about by Dr. Louiselle, if I'm saying her name correctly, we know that things such as obesity and diabetes may affect a wide variety of things in terms of how drugs are processed. I'm curious to see if that's like another avenue of, even more specificity once we know which drug to give.
Yeah, and I think that brings up an important point where the patient background could alter like how they respond to like any form of generalized or generic drug or therapeutic avenues. And that's something that I don't know if it's like very well like thought about in the clinic, because when patients don't respond, it's like seen as like, oh, like we just move on to the next thing, as opposed to trying to figure out, oh, like why didn't they respond to that particular therapy? And something we're hoping to like help clarify is that point, is that like each patient shouldn't like, I guess like there's no magic key as kind of the point is, we should, if we're going to do precision medicine, we should truly do personalized precision medicine.
I think
one question, sorry.
Yeah.
About the machine learning approach. I guess what were some considerations technically about maybe which models to use given the data structure that you're working with, what kind of informed that thought process?
So most of the time, our data is very it's not like a dynamic response type of data set, so we can't apply most models to it. Like the most we can do with the data is do clustering, correlation, or covariation type of analysis. So that's kind of what informs our analysis. Now, depending on like how robust the data is and like how much variance there is, you can apply simpler models versus if the differences are like more nuanced, then that becomes a more involved model. So for the more nuanced stuff, we do regression-based analysis, and then for the more robust or like the changes that have like higher magnitude, we do like discriminant type of analysis. So that's kind of how we're informing, depending on like how robust the data is. And I mean, like the statistical method doesn't like do much once you've acquired the data. So we also take into account like how much the biology is changing. Like the challenge is the biology sometimes doesn't change much, but whatever small tiny change there is there, like that could actually influence a lot of the systems level response. So we're taking that into account, and where possible, we kind of narrow down our analysis to what's important or what we think would be essential for that particular sample set. So we do a lot of like reduction. Great. Thank you for all the awesome questions.
All right, thank you all. Thanks so much for this wonderful presentation and the lively discussion. I feel like I learned a lot today. I'm not always in the proteomics space, but this was really cool to see, and I'm excited to see what comes next from your research. We have some, I guess, announcements. Let me share my screen. Or some outro. Sorry, I lost Zoom. But we have, again, the event feedback form. If you would like to go to that link and let us know how you did. We also have some, we have a general and member specific, I guess, survey about Comp Bio. And so if you have time to do those, that would be great. We're also starting our giving campaign for the end of the year, which will start on Giving Tuesday and end at the end of the year. And we're opening up 3 new ways to give to BWCB. You can impact our workshop fund, which will help our members participate in workshops and learn more analytic techniques like proteomics. Give to our travel fund, which will help us get our members to more conferences, and also to our honorarium fund, which will help to bring more speakers like Dr. Tamir here and sharing their research. Any other announcements, Janae?
No, and yeah, one question, and Zinga put in the chat, if you're a member, there's a separate link to the member survey specifically in your inboxes. If you have any questions about that, feel free to email us or Slack us. And then the second link I put in the chat goes directly to our donation page. If you have questions, feel free to email treasury@blackwomencompbio.org. But yeah, thank you all for your support and for coming today. It's really appreciated. Great turnout.
Yeah. All right, well, thanks again for coming. We'll see y'all next time.